Look at the color of your skin. Whatever it is, you are almost certainly wrong about where it came from. We were taught a simple story. The farther your ancestors lived from the equator, the lighter their skin became slowly over thousands of generations in order to synthesize vitamin D. The real genetics tells a different story. The color of your skin is not a slow climatic drift. It is the trace of specific migrations, specific waves of people who mixed, replaced one another, and carried genes for lighter skin into places where, in geological terms, that skin color simply did not exist the day before. Before we go any further, please subscribe to this channel. Your support genuinely helps us keep making films like this one. Thank you. Now, let's talk about a face that should not have existed, according to everything science believed for the better part of a century. In 1903, workers improving drainage at Gough's Cave in Cheddar Gorge in the English county of Somerset uncovered a nearly complete human skeleton. It would eventually be dated to approximately 10,000 years ago, making it the oldest, essentially complete skeleton of a modern human ever found in Britain. For more than a century, this individual, who came to be known inevitably as Cheddar Man, sat in museum collections as a curiosity, a set of bones with no face, no color, no particular story beyond his age and his location. Scientists assumed, reasonably enough given the prevailing model of human evolution, that whoever he was, he probably looked something like a modern Briton. Pale skin, perhaps light eyes, a face suited by 10,000 years of adaptation to the grey, sun-starved climate of Northern Europe. It is worth pausing on exactly why a discovery like Cheddar Man's genome was not possible until 2018. More than a century after his skeleton was first excavated, because the technical story behind that delay illuminates something important about how quickly this entire field has moved, and how provisional any current understanding of ancient human genetics necessarily remains. Extracting usable DNA from remains 10,000 years old presents a formidable set of technical challenges, entirely distinct from sequencing DNA from a living person. DNA degrades steadily over time, breaking into increasingly short fragments as chemical bonds within the molecule gradually fail, and by the time remains have spent 10 millennia buried in soil exposed to fluctuating temperature, moisture, and microbial activity. The surviving genetic material typically exists only as a scattered collection of short, damaged fragments, heavily contaminated with DNA from soil bacteria, fungi, and the countless other organisms that have interacted with the remains over the intervening thousands of years. Distinguishing the genuine, if severely degraded, human genetic signal from this overwhelming background contamination requires both specialized laboratory protocols designed to prevent modern human DNA from contaminating samples during handling, and increasingly sophisticated computational methods capable of reassembling millions of short, damaged fragments into a coherent, statistically confident genetic sequence. The specific techniques that made the Cheddar Man analysis possible: high-throughput sequencing technology, capable of processing millions of DNA fragments simultaneously, combined with statistical methods for authenticating genuinely ancient DNA, and distinguishing it from contamination based on characteristic patterns of chemical damage that accumulate in a predictable way over archaeological timescales, did not exist in a practical, affordable form until roughly the mid-2000s, and continued improving substantially through the following decade. Researchers working with the Cheddar Man remains in earlier decades simply lacked the tools to extract any meaningful genetic information at all. The skeleton could be dated, measured, and examined for signs of injury or disease, but its owner's actual physical appearance remained entirely inaccessible to direct scientific investigation, until the specific combination of extraction and sequencing technology available by 2018 finally made it possible. This technological trajectory matters for understanding the broader arc of this film's argument, because it means the entire body of evidence discussed here, from Cheddar Man's genome to the large-scale ancient DNA surveys spanning the Paleolithic through the Iron Age that produced the 2025 finding about the timing of skin lightening across Europe, represents a genuinely recent scientific capability, most of it developed and refined within the past 15 to 20 years. The old, simplified vitamin D drift model of skin color evolution was not overturned because researchers suddenly became more careful or more clever. It was overturned because an entirely new category of direct evidence, physically reading genetic information out of ancient human remains rather than inferring population history indirectly from patterns of variation in modern populations, became available for the first time. And that evidence turned out to tell a considerably more complicated story than indirect inference alone had been able to detect. In 2018, researchers at University College London and the Natural History Museum in London finally extracted usable ancient DNA from Cheddar Man's skeleton, a technical achievement that had simply not been possible in earlier decades, when sequencing technology could not reliably recover genetic information. This degraded, this old, this compromised by 10 millennia of burial. What they found, when they finally read the sequence, did not match the assumption at all. Cheddar Man had dark skin. Not olive, not tan, not an ambiguous intermediate tone open to interpretation. The genetic markers were unambiguous. Dark, likely approaching the pigmentation levels seen today in parts of sub-Saharan Africa. And his eyes, reconstructed from an entirely separate set of genetic markers, since eye color and skin color are controlled by largely independent genetic systems, were blue. Dark skin, blue eyes, in Britain, 10,000 years ago. This is worth sitting with for a moment, because the specific combination is what made the findings so genuinely disorienting to researchers, rather than simply a minor correction to an established picture. If skin color evolved as a slow, steady climatic adaptation, as the traditional model held, with skin gradually lightening as human populations moved into higher latitudes and lower ultraviolet exposure over the tens of thousands of years since our species first left Africa, then a population that had been living in Britain at a latitude receiving dramatically less sunlight than equatorial Africa for many thousands of years by the time Cheddar Man was alive, should already have shown meaningful movement toward lighter pigmentation. The clock, on the traditional model, had been running for a very long time. Cheddar Man's DNA suggested the clock had, in a specific and important sense, barely started. And Cheddar Man was not an anomaly, a single strange individual whose genetics happened not to match his neighbors. Subsequent analysis of ancient DNA, recovered from other Mesolithic-era human remains, found across Western Europe, from La Branya in northern Spain, from sites in Luxembourg, from multiple locations across the continent, revealed the same basic pattern again and again. Dark skin, frequently combined with light-colored eyes, blue or green. This was not the exception among Western European hunter-gatherers of this period. According to the genetic evidence now available, it was close to the rule. It is worth returning in more detail to a point raised earlier but not fully developed. The fact that Cheddar Man's blue eyes and dark skin, a combination many people find genuinely difficult to intuitively reconcile, reflects the operation of largely separate genetic systems rather than any kind of biological contradiction. Eye color in humans is determined primarily by the type and quantity of melanin present, specifically within the iris, governed substantially by variation at a small number of genes distinct from, though partially overlapping with, the broader set of genes controlling skin pigmentation. The gene most strongly associated with the specific blue versus brown eye color distinction, called HERC2, contains a regulatory region that controls the activity of a neighboring gene, OCA2, effectively acting as a genetic switch that determines how much melanin accumulates within iris tissue. A specific variant of this regulatory region, associated with reduced OCA2 activity and correspondingly reduced iris pigmentation, appears to have arisen from a single common mutational origin, with population genetic research suggesting this specific blue eye-associated variant spread from one original source individual, at some point probably between 6,000 and 10,000 years ago, and subsequently propagated widely enough across ancient European populations to account for the surprisingly high frequency of blue eyes documented among Mesolithic hunter-gatherers, including Cheddar Man himself, despite those same populations retaining predominantly dark skin pigmentation, controlled by an almost entirely separate set of genetic variants. This genetic separation between eye color and skin color explains why a combination that strikes modern intuition as unusual or even contradictory was, in fact, entirely unremarkable from a genetic perspective. There is no shared underlying mechanism requiring skin color and eye color to change in lockstep with one another. A population can carry a genetic variant favoring blue eyes, while simultaneously retaining ancestral dark skin variants at an almost entirely different, largely independent set of genes. And this is very likely exactly what occurred across Mesolithic Western Europe, the blue eye variant spreading through the hunter-gatherer population, for reasons that remain somewhat less well understood than the pigmentation story traced throughout the rest of this film. Well before the light-skin variants arrived from Anatolia, several thousand years later. Hair color pigmentation follows a broadly similar pattern of partial genetic independence from both skin and eye color, involving its own specific combination of variants, most prominently at the MC1R gene, already discussed in the context of both modern humans and Neanderthals. Along with contributions from several additional genes affecting the relative balance between eumelanin and pheomelanin, specifically within hair follicle cells. Red hair, in particular, results from a specific category of MC1R variants that substantially reduce the receptor's functional activity, shifting pigment production away from eumelanin and toward pheomelanin. The same basic biochemical mechanism, though achieved through different specific mutations, responsible for the pale skin and red hair combination. Documented in the Neanderthal MC1R variant discussed earlier in this film. The broader lesson embedded in this genetic architecture, worth stating explicitly, is that human pigmentation should never have been conceptualized as a single trait in the first place, however convenient that simplification might be for casual conversation or introductory textbook summaries. Skin color, eye color, and hair color are controlled by substantially overlapping but genuinely distinct sets of genes capable of evolving along partially independent trajectories, responding to at least somewhat different selective pressures, and combining in any given individual or population according to whatever specific mix of variants that population's particular migratory and mixing history happened to produce. Cheddar Man's dark skin and blue eyes were not a paradox requiring special explanation. They were simply what happens when two largely independent genetic systems, each with its own separate evolutionary history, happen to be examined together in the same well-preserved ancient skeleton. To understand why this matters so much, and why it required overturning a model that had dominated popular and scientific thinking about human pigmentation for decades, we need to go back to the basic biology of what skin color actually is and what problem it is generally understood to solve. Human skin color is determined, in the most immediate physical sense, by the type and quantity of a pigment called melanin, produced by specialized cells called melanocytes embedded in the skin's outer layer. Melanin exists in two primary chemical forms: eumelanin, which produces brown and black tones and offers significant protection against ultraviolet radiation, and pheomelanin, which produces red and yellow tones and offers considerably less protective capacity. The relative balance and total quantity of these two pigments, produced by essentially the same population of melanocyte cells present in every human being regardless of ancestry, accounts for the vast majority of the visible variation in human skin tone across the entire species. It is worth pausing on the underlying biology in more detail, because the mechanism involved is more intricate and more genuinely interesting than the simplified textbook version usually conveys. Melanocytes, the specialized pigment-producing cells embedded throughout the skin's outer layer, do not simply sit passively producing a fixed quantity of pigment. They respond dynamically to ultraviolet exposure, which is why human skin tans, temporarily darkening in response to sun exposure as melanocytes ramp up production in a short-term defensive response. Distinct from the longer-term genetic differences in baseline pigmentation that this film is primarily concerned with. The genetic variants responsible for the deep, permanent differences in baseline skin tone between human populations affect not how much melanin a given amount of ultraviolet exposure triggers a person's melanocytes to produce, but rather the underlying baseline quantity, type, and distribution of melanin present in the skin, regardless of recent sun exposure. The specific damage ultraviolet radiation causes to unprotected skin operates through several overlapping mechanisms. And it is worth being precise about them because they explain why the evolutionary pressure involved was genuinely severe rather than merely cosmetic. Ultraviolet radiation, absorbed directly by DNA molecules within skin cells, can cause a specific kind of chemical damage called a pyrimidine dimer, in which two adjacent bases on a DNA strand become abnormally bonded together, disrupting the cell's ability to accurately read and replicate its own genetic code. Left unrepaired or repaired imperfectly, this kind of damage accumulates over a lifetime of sun exposure and measurably increases the risk of skin cancers, including melanoma, the most dangerous and most rapidly lethal form. This cancer risk alone would represent meaningful evolutionary pressure, but the pressure operating most directly on reproductive success, and therefore most directly on the pace of natural selection, appears to have come from a different and less immediately obvious source: folate. Folate, also known as vitamin B9, is essential for DNA synthesis and cell division, and it is particularly critical during pregnancy when rapid cell division in a developing embryo depends on an adequate, uninterrupted folate supply. Folate deficiency during early pregnancy is strongly associated with neural tube defects, including spina bifida, a serious and potentially life-threatening congenital condition. Ultraviolet radiation, it turns out, directly breaks down folate circulating in the bloodstream in a photochemical reaction that occurs even in skin that shows no visible sign of sun damage. Populations with lightly pigmented skin living under intense near-equatorial ultraviolet exposure would, on this model, have faced meaningfully elevated risks of folate depletion and its associated reproductive consequences, providing a second, independent, and arguably more evolutionarily forceful pressure, favoring darker skin in high ultraviolet environments, operating alongside and reinforcing the more commonly cited cancer risk pressure. Modern medicine offers a striking, if unintentional, natural experiment that illustrates both sides of this evolutionary tension with unusual clarity. Individuals with albinism, a genetic condition resulting in little or no melanin production regardless of their broader ancestral population, face dramatically elevated skin cancer risk when living in high ultraviolet environments. A pattern documented extensively in albinism research conducted across equatorial African nations, where the condition's health consequences are measurably more severe than in populations living at higher latitudes with reduced ambient ultraviolet exposure. At the opposite extreme, individuals with naturally very dark skin living at high latitudes, particularly following migration to regions with dramatically reduced ultraviolet exposure compared to their ancestral environment, show measurably elevated rates of vitamin D deficiency compared to lighter-skinned populations native to the same region, a documented modern public health concern, particularly among immigrant populations in Northern Europe and North America, that stands as a direct, observable illustration of exactly the underlying biological tension this film has been describing, now playing out on a modern timescale far too short for further genetic adaptation to meaningfully intervene. The reason melanin exists at all, from an evolutionary perspective, connects directly to the physics of ultraviolet radiation and its complicated, double-edged relationship with human health. Ultraviolet radiation from the sun causes measurable damage to DNA within skin cells, and sustained, high exposure, the kind experienced by populations living at or near the equator under intense, nearly directly overhead sunlight for most of the year, creates elevated risk of skin cancer and, significantly for reproductive success in an evolutionary context, can damage folate, a B vitamin critical for healthy fetal development during pregnancy. High concentrations of eumelanin provide meaningful protection against this specific damage, which is why populations that evolved in regions of intense, sustained ultraviolet exposure across equatorial Africa, but also in other high UV regions of the world, generally carry genetic variants producing darker skin. But ultraviolet radiation is not simply a hazard to be defended against. It also serves an essential biological function. It triggers a chemical reaction in the skin that produces vitamin D, a nutrient required for calcium absorption, bone health, and a wide range of other physiological processes. In regions with lower ultraviolet intensity, generally, though not perfectly, correlating with higher latitudes farther from the equator, heavily melanized skin can, in principle, block enough ultraviolet radiation to meaningfully reduce vitamin D synthesis, particularly during winter months when the sun's angle further reduces the intensity of available UV radiation reaching the ground. This tension, protection against UV damage on one side, sufficient UV penetration for vitamin D synthesis on the other, is the foundation of what researchers call the vitamin D hypothesis for the evolution of lighter skin, a model that has circulated in various forms since the mid-20th century, and that, until recently, represented something close to the default explanation offered in textbooks, documentaries, and popular science writing whenever the topic of human skin color variation came up. Populations that moved into higher latitudes, the model proposed, experienced sustained selective pressure favoring lighter skin, because lighter skin allowed for more efficient vitamin D synthesis under conditions of reduced ambient sunlight. And this pressure operated gradually, consistently, generation after generation, producing the geographic gradient of skin tone that maps, roughly, onto distance from the equator across much of the modern world. It is worth tracing the history of the vitamin D hypothesis itself, because the model did not emerge fully formed, and its own history illustrates how scientific understanding of this specific question has repeatedly been revised as new evidence accumulated. Early 20th century researchers had already established a clear geographic correlation between skin pigmentation and latitude across human populations worldwide, but the specific biochemical mechanism connecting the two remained genuinely unclear for decades. The vitamin D synthesis pathway itself was not fully characterized until well into the 20th century, and it was not until 1967 that the biochemist W. Farnsworth Loomis published an influential paper in the journal Science, formally proposing that variation in human skin pigmentation had evolved primarily to balance the competing demands of ultraviolet protection and vitamin D synthesis. A framework that would go on to become the dominant explanatory model taught in textbooks and popular science writing for the following several decades. Loomis's original formulation focused primarily on the vitamin D side of this balance, framing lighter skin at higher latitudes as an adaptation specifically to prevent vitamin D deficiency and its associated health consequences, including rickets, a bone development disorder that had been documented at elevated rates in poorly nourished, sun-deprived populations in industrializing northern European cities during earlier centuries. It was only in subsequent decades, as researchers including Nina Jablonski and George Chaplin developed and refined the model further through the late 1990s and early 2000s, that the folate protection side of the equation received comparable attention, producing the more complete, two-sided evolutionary framework. Protection against folate photolysis pulling toward darker skin near the equator, protection against vitamin D deficiency pulling toward lighter skin at higher latitudes. That represents the current mainstream understanding of the basic selective pressures involved. What this history makes clear is that even the underlying biological mechanism, well before any of the ancient DNA evidence discussed throughout this film became available, was itself the product of decades of incremental scientific refinement, with earlier, simpler versions of the model gradually giving way to more complete ones as additional evidence accumulated. The specific error this film has focused on, the assumption that this biological mechanism operated as a slow, steady, population-wide drift occurring gradually and continuously since the initial human dispersal out of Africa, was, in a sense, a separate and additional assumption layered on top of an already evolving biochemical model. One that ancient DNA evidence has now shown to be considerably less accurate than researchers working with only modern population genetics had reasonable grounds to assume. The model is not wrong in its basic chemistry. The relationship between ultraviolet exposure, melanin, and vitamin D synthesis is real and well documented. What the Cheddar Man evidence and the substantial body of subsequent ancient DNA research it helped inspire revealed was that the timeline implied by treating this as a slow, steady, climate-driven gradient was fundamentally wrong. Not wrong about the end point, since modern Northern Europeans genuinely do carry, on average, much lighter skin than modern equatorial African populations, but wrong about the pace, the mechanism, and, critically, about when this transition actually occurred. Cheddar Man's genome lacked the specific genetic variants now understood to be primarily responsible for light skin in modern Europeans. Two genes in particular carry the largest share of this effect: SLC2 SLC24A5 and SLC45A2, both of which affect the internal cellular machinery involved in melanin production and distribution within skin cells. Modern populations of European descent carry the derived light-associated variant of SLC24A5 at frequencies approaching 100%, and the light-associated variant of SLC45A2 at frequencies in the range of 80 to 95%, depending on the specific population sampled. Cheddar Man and the broader population of Western European hunter-gatherers he represents carried neither. This raised an obvious question, and answering it required a fundamentally different kind of evidence than a single well-preserved skeleton could provide. If Mesolithic hunter-gatherers across Western Europe were still, 10,000 years ago, carrying predominantly dark skin-associated genetic variants, despite many thousands of years of prior residence at latitudes with substantially reduced ultraviolet exposure. When and how did the light skin variants that define the pigmentation of most modern Europeans actually arrive and spread, and eventually become the overwhelming majority pattern we observe today? The answer that has emerged from a growing body of ancient DNA research over the past several years is not a story of gradual, in-place adaptation at all. It is a story of migration, replacement, and genetic mixing occurring on a timescale, and through a mechanism considerably more recent and considerably more eventful than the old climate adaptation model implied. Beginning roughly 8,000 to 9,000 years ago, a new population began arriving in Europe from the direction of Anatolia, the region corresponding to modern-day Turkey, bringing with them not only a fundamentally different subsistence strategy, agriculture, but also a substantially different genetic profile from the hunter-gatherer populations already established across the continent. These early farming populations, migrating gradually into Europe over the following several thousand years and progressively displacing, absorbing or mixing with the existing Mesolithic hunter-gatherer inhabitants, carried meaningfully higher frequencies of the light skin-associated variants at both SLC24A5 and, somewhat later in the genetic record, at genes including TYR, another significant contributor to pigmentation variation. The migration responsible for introducing light-skin genetic variants into Europe deserves closer examination in its own right, because it represents one of the most thoroughly documented population movements in the entire ancient DNA record. And the specific evidence behind it illustrates just how detailed this kind of genetic reconstruction has become. Large-scale ancient genome sequencing projects, drawing on skeletal remains recovered from archaeological sites across Anatolia, the Aegean, and the Balkans, have established that the population responsible for introducing farming into Europe emerged from a genetically distinct source population centered in what is now central and western Turkey, carrying a genetic profile measurably different from the Mesolithic hunter-gatherer populations already established across the European continent at the time of first contact. Research led by population geneticists, including Iosif Lazaridis and colleagues, published across a series of influential papers through the 2010s, used large comparative datasets of ancient genomes to trace this Anatolian farming population's expansion into Europe in considerable geographic and chronological detail, documenting its arrival in Greece and the Balkans by approximately 8,000 years ago. Its subsequent spread along two broadly separate routes, one following the Mediterranean coastline westward, another following the Danube River corridor into Central Europe, and its progressive movement into Northern and Western Europe over the following 2,000 to 3,000 years. What these detailed genomic reconstructions reveal is not a simple, one-directional replacement of hunter-gatherers by farmers, but a considerably more complicated, regionally variable process of contact, competition, and gradual genetic mixing that unfolded differently in different parts of the continent. In some regions, genetic evidence suggests farming populations expanded with comparatively little admixture from existing hunter-gatherer inhabitants, at least initially. In others, meaningful mixing occurred relatively early and continued over subsequent centuries. Genetic contributions from the original Mesolithic hunter-gatherer populations, including the dark skin-associated genetic profile carried by individuals like Cheddar Man, did not disappear from the European gene pool following the arrival of farming populations. They persisted in varying proportions across different regions, continuing to contribute to the ancestry of subsequent European populations for thousands of years afterward, which is precisely why the transition toward predominantly light skin, when eventually mapped out across the full chronological span of ancient DNA evidence, appears as the gradual, uneven, centuries-spanning process the 2025 research describes, rather than a single clean replacement event occurring at one identifiable moment in time. A further genetic contribution complicates the picture, even beyond the initial hunter-gatherer and farmer populations already discussed. Beginning roughly 5,000 years ago, a third major population movement swept into Europe from the Pontic-Caspian steppe, the vast grassland region north of the Black and Caspian Seas, spanning parts of modern Ukraine and southern Russia. A population associated archaeologically with the Yamnaya culture and genetically linked to the subsequent spread of Indo-European languages across much of Europe and South Asia. This steppe-associated population carried its own distinct genetic profile, including its own contribution to the ongoing pigmentation story, and its arrival and subsequent mixing with the existing farmer and hunter-gatherer descended populations already present across Europe added yet a third genetic layer to the increasingly complex ancestry of the populations that would eventually become recognizable genetically as the ancestors of most modern Europeans. The light skin variant story traced throughout this film, in other words, does not involve simply two populations meeting once and mixing. It involves at minimum three genetically distinct major populations: Mesolithic hunter-gatherers, Anatolian-derived Neolithic farmers, and Bronze Age steppe pastoralists, arriving in sequence over a span of several thousand years, each contributing a different genetic profile, mixing with the populations already present to different degrees in different regions, and collectively producing the genuinely intricate, regionally variable pattern of pigmentation genetics that ancient DNA research has only recently become capable of reconstructing in this level of detail. This is the mechanism that a landmark ancient DNA study, examining genetic samples spanning the full arc from the Paleolithic through the Mesolithic, Neolithic, Copper Age, Bronze Age, and into the Iron Age, was designed specifically to trace in detail published research whose findings, reported widely in late 2025, produced a headline conclusion that would have seemed almost unbelievable to researchers working with the tools available even a decade earlier. Most Europeans retained dark skin until as recently as 3,000 years ago. 3,000 years. Not the 10,000 years separating us from Cheddar Man, and not the tens of thousands of years separating modern Europeans from the original out-of-Africa migration that populated the continent in the first place. 3,000 years, a span of time comfortably within recorded human history, overlapping directly with the era of ancient Egyptian temple construction, Mesopotamian legal codes, and the Bronze Age civilizations of the Eastern Mediterranean that appear, however briefly, in most standard history curricula. While these civilizations were already flourishing, keeping written records, building monumental architecture that still stands today, a substantial proportion of the population living across Europe still carried the dark or intermediate skin pigmentation inherited from earlier hunter-gatherer and early farming ancestors. The specific pattern the researchers documented was not a clean, simple transition either. Working through their sequence of ancient samples chronologically, black dots on their published distribution maps marking Paleolithic individuals, blue marking Mesolithic, yellow marking Neolithic, green marking Copper Age, orange marking Bronze Age, purple marking Iron Age, the data showed something considerably messier than a steady march from dark to light. The shift toward lighter pigmentation was not linear in time, and it was not uniform in geography. Populations in different regions of Europe show meaningfully different trajectories, different rates of change, and different final compositions, depending on exactly which migrating populations reached them, when, and in what proportion they mixed with the people already living there. A separate but closely related line of research reinforces just how genuinely non-simple this genetic history turns out to be once researchers look closely enough. Rather than representing a single trait selected for and spread uniformly through natural selection acting independently in every population exposed to reduced ultraviolet radiation, the researchers behind this body of work have identified evidence of what geneticists call adaptive gene flow, meaning that at least some of the specific genetic variants responsible for lighter pigmentation appear to have entered the broader European gene pool not through independent natural selection arising separately, population by population, but through interbreeding between different, genetically distinct populations that had already, for reasons specific to their own separate histories, evolved these variants somewhere else, at some earlier point, under conditions we do not yet fully understand. This distinction matters considerably more than it might initially appear to. A trait spreading through independent, parallel natural selection in multiple separate populations each responding to a broadly similar environmental pressure, tells one kind of evolutionary story. A story about convergent adaptation, about environment reliably producing similar outcomes wherever similar conditions exist. A trait spreading primarily through migration and interbreeding, carried by specific populations moving into new territory and mixing with populations already living there, tells an entirely different kind of story, one dependent on the specific, contingent, and in principle avoidable historical fact of which populations happen to move where and when and how extensively they mixed with their new neighbors rather than remaining genetically separate. The first story is about biology responding predictably to environment. The second is about history, messy, particular, difficult to predict human history, leaving a permanent trace in the human genome. There is a second thread in this story that deserves equal attention, because it adds a genuinely strange complication to the picture, one that becomes visible only when we widen our lens beyond Homo sapiens entirely, to include the Neanderthals, the closely related, now extinct human species that occupied Europe and Western Asia for hundreds of thousands of years, before eventually disappearing from the fossil and genetic record roughly 40,000 years ago. In 2007, a research team successfully extracted and sequenced a fragment of a specific gene, MC1R, the Melanocortin 1 receptor gene, a critical regulator of the pigmentation pathway in humans, and in fact, across the broader animal kingdom, from birds to mammals, from two separate Neanderthal specimens. What they found was a mutation, a specific variant of this gene that had never once been observed in any of the roughly 3,700 modern human genetic samples the researchers used for comparison at the time. Laboratory testing of this specific Neanderthal variant confirmed that it meaningfully reduced the functional activity of the MC1R protein, precisely the kind of functional change known from extensive study of modern human populations to produce paler skin and, in some cases, red hair when it occurs in Homo sapiens. The implication of this finding is worth stating plainly, because it directly overturns an assumption that might otherwise seem like the obvious, parsimonious explanation for any shared trait between Neanderthals and modern humans. Given that we know these two species interbred at various points, where their ranges overlapped, leaving a measurable genetic legacy, somewhere between 1 and 4% of the genome of most present-day non-African humans, still present in living populations today. Given that legacy, a reasonable first assumption might be that any light pigmentation genes shared between Neanderthals and modern Europeans arrived in our species through exactly that interbreeding, the same pathway responsible for several other well-documented Neanderthal genetic contributions to the modern human genome. That is not what happened here. The specific MC1R variant identified in Neanderthal DNA is not the same variant responsible for reduced pigmentation in modern humans. It is a genuinely distinct mutation, arising independently within the separate evolutionary lineage that produced Neanderthals, entirely disconnected from the various distinct mutations that separately arose within the Homo sapiens lineage, and that are responsible for the pale skin and red hair variants observed in modern human populations today. Modern Europeans, subsequent and more comprehensive genetic research has confirmed, did not inherit their lighter pigmentation from Neanderthal ancestors, despite the genuine genetic mixing that occurred between the two species. The light skin variants present in modern Homo sapiens populations evolved entirely separately within our own species at a different point in time through different specific mutations arising in different genes. What this means, considered directly, is that pale skin appears to have evolved independently, at least twice, in two closely related but genetically and evolutionarily distinct human lineages, occupying broadly overlapping geographic ranges and broadly similar environmental conditions across Ice Age Europe, without either lineage borrowing the relevant genetic solution from the other, despite ample documented opportunity to do so through interbreeding. This is precisely the kind of finding that should make anyone thinking carefully about the story of human pigmentation pause and reconsider how confidently a single, tidy narrative can be applied to it. Convergent evolution, separate lineages independently arriving at similar solutions to similar environmental problems, is a well-documented phenomenon throughout the broader history of life on Earth. Finding it operating between Neanderthals and modern humans on a trait as visible and as culturally loaded as skin pigmentation adds yet another layer of genuine complexity to a story that popular science had for a very long time, told as though it were considerably simpler than the underlying evidence actually supports. This deep evolutionary history connects directly to measurable public health patterns observed in modern populations today, and examining that connection helps ground the more abstract genetic and archaeological material already discussed in something immediately relevant to living people. Skin cancer incidence rates, tracked extensively across national health systems worldwide, show a strong and well-documented correlation with both baseline population pigmentation and geographic patterns of migration that have occurred far too recently for further genetic adaptation to have meaningfully occurred. Australia and New Zealand, populated predominantly by descendants of comparatively light-skinned European migrants, who arrived within the past 2 to 3 centuries and settled at latitudes receiving significantly more intense ultraviolet exposure than their ancestral homelands, report some of the highest melanoma incidence rates recorded anywhere in the world, a documented public health consequence of population movement, occurring on a timescale of mere centuries, far too rapid for the kind of gradual genetic adaptation traced throughout this film to have had any meaningful opportunity to operate. The mismatch between a population's evolved baseline pigmentation and its current environment, in other words, is not merely a historical curiosity relevant to interpreting ancient skeletons. It remains an active, measurable, ongoing feature of modern human health, playing out in real time across populations that have moved geographically far faster than their genomes have been able to follow. The opposite side of this same underlying tension shows up with comparable clarity in modern vitamin D deficiency statistics. populations with comparatively dark baseline skin pigmentation who have migrated within the past several generations to regions with significantly reduced ambient ultraviolet exposure. Documented extensively among South Asian, African, and Middle Eastern immigrant communities settled in Northern Europe and parts of North America, show measurably elevated rates of vitamin D deficiency compared to lighter-skinned populations native to the same regions, a pattern replicated across numerous public health surveys conducted over the past 2 decades. This is, in effect, a live demonstration of exactly the selective pressure this film has spent considerable time describing in its ancient, evolutionary form, now playing out over a handful of human generations rather than the many thousands of generations required for genuine genetic adaptation, and consequently addressed today primarily through dietary supplementation and public health guidance rather than through any expectation of further evolutionary change occurring on a practically relevant timescale. These modern patterns matter for understanding the broader argument this film has developed, because they demonstrate that the underlying evolutionary logic connecting ultraviolet exposure, melanin, and human health was never merely an interesting historical explanation for a pattern that had already finished unfolding thousands of years ago. It describes a genuinely ongoing biological relationship, one that continues to shape measurable health outcomes today, precisely because human migration across the entire span of our species' history and continuing into the present has always moved dramatically faster than genetic adaptation is capable of following. The mismatch that produced Cheddar Man's unexpectedly dark skin in Mesolithic Britain, and the mismatch that produces elevated melanoma rates among light-skinned populations in modern Australia, are, at the deepest biological level, expressions of exactly the same underlying phenomenon, separated only by which direction the mismatch happens to run, and by how many thousands of years have or have not yet passed since the relevant migration occurred. There is a third case worth examining alongside Cheddar Man's Western Europe and the independently evolved Neanderthal variant, because it adds a genuinely different kind of complication to the picture, and because it involves one of the oldest, most continuously distinct human lineages currently known to genetic science. The Khoisan peoples of southern Africa, including the San, sometimes historically referred to by the now outdated term Bushman, and the closely related Hwekhawe pastoralist populations represent, according to extensive population genetic research, one of the deepest and most anciently distinct branches of the entire modern human family tree, with genetic evidence indicating their lineage diverged from other human populations somewhere between 100,000 and 150,000 years ago. Tens of thousands of years before the migrations out of Africa that eventually populated Europe, Asia, and the Americas. In evolutionary terms, the Khoisan are, in a meaningful sense, more distantly related to other African populations than many of those other populations are to Europeans or East Asians. A reflection of just how much genetic diversity exists within Africa itself, often underrepresented in genetic research relative to the comparatively narrow slice of human diversity carried by populations descended from the more recent out-of-Africa migrations. And the Khoisan, notably, have measurably lighter skin than most other sub-Saharan African populations. A long-recognized observation that researchers had, for years, tentatively attributed to local adaptation, reasoning that the specific environment of the southern Kalahari and surrounding regions might present somewhat different ultraviolet exposure conditions than equatorial Africa, potentially favoring somewhat lighter pigmentation through the same basic vitamin D and folate selective framework already discussed. A study published in the Proceedings of the National Academy of Sciences in December of 2018 revealed something considerably more specific and considerably stranger than simple local adaptation. Examining the genetic basis of this lighter pigmentation in Khoisan populations, the research team identified the same gene already discussed extensively throughout this film, SLC24A5, the single largest genetic contributor to light skin in European populations. And critically, when the researchers examined the specific stretch of DNA surrounding the light-associated variant of this gene in Khoisan individuals who carried it, they found it was virtually identical to the corresponding stretch of DNA in Europeans carrying the same variant, a degree of genetic similarity far too precise to have arisen through two entirely separate, independent mutations occurring by chance in two populations that had been genetically isolated from one another for over 100,000 years. The only plausible explanation, confirmed through subsequent detailed population genetic modeling, was that this was the identical mutation, originating once and then spreading. Further analysis traced its specific path. The light-associated SLC24A5 variant appears to have arisen somewhere in the vicinity of Europe or the Near East within roughly the past 10 to 35,000 years, spread from there into Eastern African pastoralist populations through the same broad patterns of migration and admixture responsible for spreading agriculture and pastoralism across the African continent, and from Eastern Africa, carried by migrating pastoralist populations bringing domesticated sheep and goats southward, arrived among the ancestors of the Khoisan as recently as roughly 2,000 years ago. A genuinely startling figure, representing barely more than 1% of the Khoisan lineage's documented history as a genetically distinct population. Once introduced through this comparatively recent interbreeding event, the variant then spread rapidly through the Khoisan population under strong local positive selection, rising from what was very likely a rare, newly introduced variant to its present, measurably elevated frequency within a span of time. Researchers explicitly describe as one of the more striking documented examples of rapid, ongoing human evolutionary adaptation in the archaeological and genetic record. This case matters enormously for the broader argument this film has been building, because it represents something distinct from both of the other examples already discussed. It is not independent convergent evolution, as with the separate Neanderthal MC1R mutation. It is not a comparatively gradual, still ongoing regional mixing process, as with the broader European transition traced through Cheddar Man and the subsequent Anatolian farming migration. It is instead the exact same specific genetic variant, the identical mutation, traveling an extraordinary geographic distance from its likely origin near Europe or the Near East, through Eastern Africa into the far southern reaches of the African continent, and arriving among a population whose ancestors had already been genetically distinct for well over 98,000 years by the time it finally showed up, arriving so recently and spreading so rapidly once it did that its signature remains sharply, unmistakably visible in the genome of Khoisan individuals living today. Considered together, these three cases, independent convergent mutation in Neanderthals, gradual regional population mixing across Mesolithic and Neolithic Europe, and the rapid, recent, long-distance transplantation of a single specific variant into one of the oldest human lineages on the planet, demonstrate that there is no single evolutionary story capable of accounting for the full pattern of light skin pigmentation across human populations worldwide. There are at least three genuinely different processes, operating on different timescales, through different mechanisms, in different populations, that happen to converge on broadly similar outward physical results. Skin color, examined this closely, turns out to be less like a single evolutionary answer and more like a collection of several genuinely separate evolutionary case studies that happen to share a superficial resemblance. One final population case deserves mention before this film's closing argument, because it adds yet another data point to the same broader pattern, drawn from a part of the world rarely discussed, alongside the European and African cases already covered in detail. Population genetic research examining pigmentation variation across South Asia found something that, by this point in the film, should feel familiar rather than surprising. The same light-associated variant of SLC24A5, responsible for the largest share of light skin in both Europeans, and, as already discussed, in Khoisan populations who received it through comparatively recent African migration, is also present at substantial frequency across South Asian populations, contributing to the considerable range of skin pigmentation documented across the Indian subcontinent. A 2013 study published in PLOS Genetics, examining the specific genetic sequence surrounding this variant in South Asian populations, compared to the corresponding sequence in Europeans, found the same pattern already established in the Khoisan case. The surrounding DNA was similar enough across a long enough stretch to indicate the variant shares a common, comparatively recent origin between the two populations, rather than having arisen independently through two separate mutations. Researchers describe this specific genetic relationship using the technical term identity by descent, meaning the shared DNA sequence reflects genuine common ancestry at that specific genetic location, inherited from a shared source population rather than coincidental similarity produced by chance. This finding places South Asia as a third documented recipient of the same specific genetic variant already traced from its likely origin near Europe or the Near East, through Eastern Africa and into the Khoisan populations of Southern Africa, now also documented spreading eastward into the Indian subcontinent, through a separate, though likely broadly contemporaneous, pattern of ancient migration and population mixing associated with the complex demographic history of South Asia, itself involving multiple documented waves of migration from Iran-related agricultural populations and steppe-associated populations arriving from Central Asia over the past several thousand years, mixing to different degrees with earlier South Asian hunter-gatherer populations to produce the genetically diverse, regionally variable population structure documented across the subcontinent today. Considered alongside the European and Khoisan cases already discussed in detail, the South Asian evidence completes a genuinely striking picture. A single specific genetic mutation, very likely arising once in one ancestral population, at some point probably within the past 10 to 35,000 years, subsequently spreading through at least 3 genuinely separate, geographically distant recipient populations: Europeans, Southern African Khoisan, and South Asians, through at least 2, and very likely 3, largely independent episodes of migration and interbreeding, each occurring on its own separate timeline, driven by its own separate specific historical circumstances, and each subsequently subject to its own separate pattern of local natural selection favoring the variant spread once introduced. It is worth pausing, too, on what this specific pattern of shared migrating variants implies about the broader relationship between different human populations across the planet, because it runs directly counter to another common but genetically inaccurate assumption: that populations separated by large geographic distances and by the visible physical differences that sometimes accompany that separation must necessarily also be separated by correspondingly deep, ancient genetic divergence at every level of their biology. The SLC24A5 case demonstrates precisely the opposite. Europeans, Khoisan, and South Asians, 3 populations whose modern popular image often emphasizes their visible differences and whose broader genetic ancestries do, in most respects, reflect genuinely distinct and often quite deep evolutionary separations, nonetheless share, at this one particular genetic location, a degree of similarity so precise that it can only be explained by direct, comparatively recent common inheritance. A single trait, examined at the level of the specific gene responsible for it, can tell a story of connection and shared recent history, even between populations whose broader genomes and whose broader evolutionary trajectories diverged from one another tens of thousands of years earlier. This is, in miniature, exactly the kind of complexity that population genetics research has increasingly revealed, sits beneath almost every visible human physical trait, once it is examined with sufficiently precise modern tools. A complexity considerably richer, considerably more interconnected, and considerably more resistant to simple categorical thinking than intuition, unaided by genetic evidence, would ever have predicted on its own. This is, in the most literal sense available to population genetics, a single evolutionary innovation traveling the world through human migration, rather than a trait independently reinvented wherever environmental conditions happened to favor it. It stands in direct informative contrast to the separate Neanderthal MC1R mutation and the separate East Asian pigmentation genes already discussed, both of which represent the opposite pattern. Genuinely independent evolutionary invention, arising separately in separate lineages, achieving broadly similar outward results through entirely different underlying genetic mechanisms. Between these two patterns, a single mutation spreading widely through migration on one hand, independent parallel invention on the other, sits essentially the entire genetic history of human skin pigmentation, currently documented by modern population genetics, a history considerably richer, considerably more geographically interconnected, and considerably less reducible to any single simple explanatory principle than the popular version of this story has generally allowed. Taken together, what does this accumulated evidence, Cheddar Man's unexpected combination of dark skin and blue eyes, the broader pattern of persistently dark-skinned Mesolithic populations across Western Europe, the migration-driven rather than climate-driven spread of light-skinned genetic variants beginning roughly 8,000 to 9,000 years ago, the genuinely non-linear and geographically uneven pace of that spread continuing well into the historical era, the documented role of adaptive gene flow moving already evolved variants between populations through interbreeding rather than independent selection, and the entirely separate, independently evolved pale skin mutation found in Neanderthal DNA, actually tell us about how human skin color came to look the way it does today. It tells us, first and most directly, that the old model, skin gradually and steadily lightening as human populations migrated away from the equator, driven by a slow but consistent selective pressure, favoring efficient vitamin D synthesis under reduced ultraviolet exposure, was not describing a fiction. The underlying biology of that model remains accurate. Ultraviolet exposure genuinely does correlate broadly with skin pigmentation across global populations. And vitamin D synthesis genuinely is affected by the interaction between melanin concentration and available ultraviolet radiation. But the model, as popularly told, badly mischaracterized the timeline, the mechanism, and the underlying process by which this outcome was actually achieved. It was not a slow, gradual population-by-population drift occurring steadily over the tens of thousands of years since modern humans first arrived in Europe. It was a comparatively rapid, historically contingent, migration-driven replacement and mixing event, occurring in earnest only within the last 8,000 or 9,000 years, continuing in an uneven, regionally variable pattern for thousands of years afterward, and reaching anything resembling its modern distribution only within the last 3,000 years, a period of time so recent in evolutionary terms that it overlaps directly with the earliest chapters of written human history. This reframing carries a broader implication worth making explicit, because it changes something important about how we should think about the relationship between visible human physical traits and deep evolutionary time. Skin color is, for obvious historical and social reasons, one of the most culturally significant physical traits any human being carries, freighted with an enormous weight of meaning. That has nothing to do with its actual underlying biology. Part of what makes the genuine scientific story so worth telling accurately is that it directly undermines a specific kind of intuitive, folk biological reasoning that has done real historical damage. The assumption that visible pigmentation differences between human populations reflect deep, ancient, essentially fixed biological categories, stable and unchanging across vast stretches of evolutionary time. The actual genetic evidence shows something close to the opposite. Skin color, across the specific case of European populations that happens to be the best studied and most thoroughly documented example currently available, turns out to be a comparatively recent, historically contingent, still actively shifting characteristic. The visible surface trace of a genuinely complicated sequence of migrations, population replacements, and genetic mixing events, several of which occurred well within the span of recorded human history, rather than the ancient, immutable marker of deep biological difference. It has so often, and so wrongly, been assumed to represent. There is one more dimension of this story worth exploring before we close, because it extends the argument beyond Europe specifically and toward a broader, more genuinely global understanding of how skin pigmentation varies across the entire human species. An understanding considerably more intricate than the single north-south light-to-dark gradient that dominates most casual popular thinking on the subject. The genetic architecture underlying skin pigmentation across the full breadth of human genetic diversity worldwide involves a considerably larger and more varied set of genes than the handful most heavily studied in the specific context of European population history. A significant 2024 review published in the journal Molecular Ecology, synthesizing research across multiple human populations and multiple methodological approaches, identifies at least 7 genes making substantial, independently documented contributions to the lightening of skin color, specifically within European populations alone. SLC24A5, SLC45A2, TYRP1, ASIPMC1R, and IRF4, each affecting a different stage of the complex biochemical pathway responsible for melanin production, transport, and deposition within skin cells. This is, notably, a considerably longer and more complicated list than the 2 or 3 genes most commonly mentioned in simplified popular accounts of the subject. And it understates the full complexity further, because different genetic variants, in different combinations, contribute to lighter pigmentation in other populations around the world through entirely separate evolutionary pathways. Populations in East Asia, for instance, have independently evolved lighter skin pigmentation relative to their more equatorial ancestral populations. But through a genetic pathway substantially different from the one responsible for lighter pigmentation in Europeans, involving different specific variants at different genes, arising through an independent evolutionary history entirely disconnected from the European story traced through Cheddar Man, the Anatolian farming migration, and the specific variants of SLC24A5 and TYR discussed throughout this film. This is convergent evolution operating again, at global scale this time, rather than simply between two closely related human lineages occupying the same continent. Multiple, genuinely separate human populations, independently arriving at broadly similar outward physical outcomes. Lighter skin pigmentation suited to reduced ultraviolet environments through entirely different underlying genetic mechanisms, evolving on entirely separate timelines in response to broadly similar but not identical environmental pressures. The East Asian case, mentioned briefly earlier, deserves a more detailed examination in its own right, because the specific genetic evidence here provides yet another independent confirmation of the same broader pattern. Convergent physical outcomes arising from genuinely separate underlying genetic mechanisms. Population genetic research, examining the light-associated variant of SLC24A5, the same gene responsible for the largest share of light pigmentation in both Europeans and, as just discussed, in Khoisan populations who received it through comparatively recent migration, has found that this specific variant is essentially absent in East Asian populations, present at frequencies close to zero across the large-scale population genetic surveys conducted to date. East Asian populations, despite carrying meaningfully lighter average skin pigmentation than their more southerly ancestral populations, achieved this outcome through an almost entirely separate set of genetic variants, most prominently involving a specific variant of a gene called OCA2, a gene more commonly associated in popular science writing with its role in eye color variation, but which also makes a substantial, independently documented contribution to skin pigmentation, specifically within East Asian populations, along with additional contributions from variants in genes including DCT and MFSD12, identified through large-scale genome-wide association studies conducted specifically within East Asian population cohorts over the past decade. This is convergent evolution operating at a genuinely global scale, and it is worth being precise about what that specifically means in this context. Convergent evolution describes a situation in which separate, independently evolving lineages arrive at similar physical solutions to similar environmental problems without inheriting the specific genetic solution from a shared source. The classic textbook examples typically involve entirely different species, the wing of a bat and the wing of a bird, evolved through entirely separate evolutionary pathways, despite serving a similar aerodynamic function. Finding a comparably clean example of convergent evolution operating within a single species, between different human populations that share the overwhelming majority of their genome, and diverged from common ancestors a comparatively short evolutionary time ago, is considerably less commonly discussed, but no less genuine an example of the same underlying evolutionary principle. European and East Asian populations independently arrived at lighter skin pigmentation, well suited to their respective higher latitude environments, through almost entirely separate sets of genetic mutations, arising independently at different genes at different points in time, in populations that had already been geographically and genetically separated from one another for tens of thousands of years by the time either lightening process began in earnest. The practical upshot of this pattern, considered across all of the populations and mechanisms discussed throughout this film, is that human skin pigmentation cannot be meaningfully reduced to a single gene, a single mutation, or a single evolutionary event, however tempting that kind of simplification might be for the purposes of a tidy popular explanation. It is, instead, the accumulated product of a genuinely large number of at least partially independent genetic events, some involving new mutations arising locally and spreading through direct natural selection, some involving existing variants carried into new populations through migration and interbreeding, and then locally selected for, occurring across multiple separate human populations on multiple separate continents, across a timescale stretching from tens of thousands of years ago to as recently as 2,000 years ago. That happen when the story is compressed into a brief popular summary to look considerably simpler and more unified than the underlying genetic evidence actually supports. This global pattern reinforces, rather than complicates, the central argument this film has been building toward. Human skin color, considered honestly and in light of the full weight of currently available genetic evidence, was never a single, simple, universally applicable evolutionary story unfolding identically and gradually across every human population as it moved into a new environment. It was, and remains, a genuinely complex mosaic, the accumulated, still partially unfolding result of dozens of separate genetic variants, arising independently in different populations at different points in time, spreading through mechanisms ranging from straightforward local natural selection to large-scale migration and interbreeding between genetically distinct populations, occasionally converging on broadly similar outward appearances through entirely separate underlying biological pathways, and, in the specific and now well-documented case of Western Europe, reaching anything resembling its familiar modern distribution only within the last 3,000 years. A conclusion that would have seemed, to most researchers working even a single decade ago, genuinely difficult to believe. The face reconstructed from Cheddar Man's skeleton, dark-skinned, blue-eyed, staring out from museum displays and countless subsequent news articles with an expression that reconstruction artists can only approximate, has become, almost by accident, one of the more effective single pieces of evidence available for communicating just how badly the popular, simplified version of this story diverges from what the actual genetic record shows. He was not an anomaly. He was, as far as the currently available evidence can determine, closer to the norm for his time and his place. a Western European hunter-gatherer whose appearance would very likely surprise most people alive today, who assume, understandably but incorrectly, that the physical appearance of ancient Europeans must have closely resembled the physical appearance of modern Europeans. It did not. And the specific, documented reasons why it did not: migration rather than drift, mixing rather than isolation, independent convergent mutation rather than shared inheritance, a timeline measured in mere thousands rather than tens of thousands of years, collectively tell a story about human variation considerably stranger, considerably more recent, and considerably more historically contingent than the one most of us were taught. This brings us to a conclusion that extends beyond the specific genetic mechanisms this film has traced, into territory that carries genuine social and historical weight. And it is worth stating directly and carefully rather than leaving implicit. The concept of biologically distinct human races, understood as discrete, ancient, internally uniform categories with deep evolutionary roots, stretching back tens of thousands of years, has never had solid grounding in the underlying population genetics of our species. And the specific evidence traced throughout this film adds a particularly vivid, concrete illustration of exactly why. If the light skin now closely associated, in popular imagination, with European ancestry did not exist in anything resembling its modern distribution until roughly 3,000 years ago, a period of time so recent that it post-dates the construction of the Great Pyramid of Giza, post-dates the earliest Mesopotamian writing systems, post-dates the founding of the earliest cities in the Indus Valley. Then treating this specific, comparatively recent, historically contingent trait as though it were an ancient, fixed, deeply rooted marker of essential biological difference is not simply an oversimplification. It is a direct factual error, refuted by the genetic evidence now available from the actual ancient human remains involved. What unites all of the specific mechanisms this film has traced is not their similarity to one another, but their shared dependence on history rather than biology alone, on which populations happen to move where and when, rather than on any deep, fixed division between human groups. Consider finally what all of this means for the simple act of looking at another person's face and drawing conclusions from it, something every human being does, constantly, almost entirely without conscious thought, and has done for as long as our species has existed. The genetic evidence assembled across this film does not suggest that visible human variation is meaningless or illusory. It suggests something more specific and, in its own way, more interesting. That the visible variation we notice most readily, skin tone chief among it, reflects an unusually recent, unusually mobile, and unusually migration-dependent layer of human genetic history, sitting on top of a much older, much more slowly changing genome shared overwhelmingly in common across the entire species. Reading deep, ancient, fixed biological categories into a trait this recent and this mobile was always going to be a mistake. The ancient DNA evidence has simply made that mistake impossible to sustain any longer. None of this is intended to suggest that human populations are genetically identical, or that population-level genetic variation does not exist and does not matter for legitimate scientific and medical purposes. The entire body of research traced throughout this film depends on the real, measurable, well-documented existence of genetic variation between human populations. And dismissing that variation entirely would be just as scientifically inaccurate as overstating its depth and antiquity. What the evidence does support, clearly and repeatedly, is that the specific trait most culturally weighted, most visually immediate, and most historically consequential in shaping how different human populations have treated one another across the more recent chapters of human history, skin color, turns out to be, on close genetic examination, one of the most recent, least stable, and most demonstrably migration-driven traits currently documented in the human genome. Its modern distribution having crystallized into anything resembling its current form only within the last few thousand years. A genuinely brief moment near the very end of a story stretching back roughly 300,000 years to the origin of our species as a whole. It is also worth noting how quickly this specific area of research continues to move, because the pace of discovery traced throughout this film shows no sign of slowing. The 2018 Cheddar Man analysis, the 2018 Quasan SLC24A5 study, and the late 2025 large-scale ancient DNA survey establishing the 3,000-year timeline for European pigmentation change were all published within a span of less than 8 years, each building directly on techniques and datasets that simply did not exist for the researchers who came before them. Ancient genome databases continue to expand rapidly, with new skeletal samples recovered, sequenced, and added to comparative datasets on an ongoing basis across research institutions worldwide, meaning the specific chronology and mechanism described in this film represents a current, well-evidenced best understanding rather than any kind of final, permanently settled account. Future discoveries may well refine the specific dates involved, identify additional migration events not yet fully characterized, or reveal further genetic complexity in populations not yet studied with comparable depth. What is unlikely to change, given how consistently the evidence has pointed in this direction across multiple independent lines of research, is the core finding this film has been built around: that human skin color is a considerably more recent, more migration-dependent, and more genuinely complicated trait than the simplified version of the story most of us grew up hearing. This is worth remembering the next time a popular headline announces some settled ancient fact about human origins with more confidence than the underlying evidence can actually support. Cheddar Man's reconstructed face, dark-skinned, blue-eyed, staring out from behind museum glass in London, entirely unaware of how thoroughly his genome would eventually complicate a story that had gone essentially unquestioned for the better part of a century, stands as a genuinely useful reminder of exactly how provisional and exactly how recent our confident-sounding accounts of deep human history actually are. The next major ancient genome, recovered from the next well-preserved skeleton, sequenced with whatever technology exists a decade from now, may well complicate this picture further still. That is not a weakness in how this particular scientific story has been told. It is close to the entire point of telling it honestly.